We will give a presentation about opsonization.
First, I explain the definition of opsonization. Opsonization is a process where the body puts a tag on a pathogen. This tag is made by things like antibodies or complement proteins. When a pathogen gets this tag, our immune cells can find it more easily. These immune cells then grab the pathogen, take it inside, and break it down. In short, opsonization means tagging pathogens so our body can find them and clean them up quickly.
Next, I will explain the biological role of opsonization. First, opsonization increases the efficiency of phagocytosis. Phagocytes have some receptors that can recognize the pathogen directly, but these receptors are limited in variety and generally show only modest affinity. In contrast, phagocytes express Fc receptors at very high density. Among antibodies, the Fab region is antibody-specific, but the Fc portion has a shared structure across all IgG molecules. Because of this shared Fc structure, phagocytes can bind to many different pathogens efficiently through a common set of receptors. As a result, once antibodies coat the pathogen surface, phagocytosis is dramatically enhanced. Moreover, opsonization is essential for the phagocytosis of certain pathogens. A classic example is encapsulated bacteria. Their capsule consists of smooth, hydrophobic polysaccharide layer that physically block pattern recognition by receptors. As a result, phagocytes can barely recognize these organisms in their native state, and phagocytosis is almost completely absent. On the other hand, antibody can recognize fine molecular structure as epitope. They can therefore bind even to encapsulated bacteria. As a result, the elimination of these bacteria requires antibody-mediated opsonization.
Next, we describe examples of opsonization. First, in the case of Staphylococcus aureus, IgG antibodies bind to specific antigens on the bacterial surface via their Fab regions. The Fc regions then stick out and are recognized by Fc gamma receptors on neutrophils and macrophages, which greatly enhances phagocytosis. Second, for E. coli, the complement component C3b can be deposited on the bacterial surface. This C3b is recognized by complement receptors, such as CR1 on phagocytes, again promoting efficient uptake and destruction of the bacteria.